Hello everyone, welcome to my YouTube channel Biotech-India. Today I'll be covering the first cell component, that is cell wall. So let's get started. Cell wall is first observed by Robert Hooke in 1965. In 1804, Carl Rudolphi and J.H.F. Ling proved that cells have independent cell wall. It's a tough covering which surrounds the cell membrane, which provides protection and distinct shape to the cell. These are the different cell walls. First is bacterial cell wall, which is composed of mucopeptide and muramic acid. Fungal cell wall, the main component is chitin in fungal cell wall. And these are the plant cell wall is made up of these many different components like cellulose, hemicellulose, protein, xylan. So we'll be starting off with the structure of bacterial cell wall. So bacterial cell wall is composed of mainly of peptidoglycan layer. The peptidoglycan is also known as murine, is a polymer which consists of sugar and amino acids that forms a mesh-like structure, as you can see here, outside the cell membrane of most of the bacteria, which forms a cell wall. It's composed of two sugar derivatives, N-acetylglucosamine and N-acetylmuramic acid, and a small number of amino acids consist of alanine, glutamic acid, lysine or diaminopalmic acid, and glycine. The sugar component consists of altering residue of beta-1,4 linked N-acetylglucosamine and N-acetylmuramic acid. So basically NAG and NAM is attached by beta-1,4 bond. These subunits, which are related to glucose in their structure, are covalently joined to one another and form a glycan chain. So you can see this is a glycan chain which is forming. Each NAM residue carries a short peptide chain of 4 amino acid residue, which is also known as a tetrapeptide, which consists of L-alanine, D-glutamic acid, L-lysine, and D-alanine. The peptide chain can be cross-linked to the peptide chain of another strand, forming a peptidoglycan, as you can see here, the structure basically. So this is all about the bacterial cell wall. So depending upon the different type structural changes in the cell wall of the bacteria, there are two types of bacteria: gram-positive bacteria and gram-negative bacteria. Gram-positive bacteria contains a large amount of teichoic acid. In gram-positive bacteria, the cross-linkage is usually by peptide in interbridge. The kinds, so this is a peptide interbridge which is forming here. And the kinds and the numbers of cross-linking amino acids varies from type of the bacteria. So this amino acid in the tetrapeptide may varies on different types of bacteria. While in gram-negative bacteria, the most important feature in gram-negative bacteria is the presence of its outer membrane, which is just lies outside the peptidoglycan layer. So this is your cell wall, gram-negative cell wall, and there is one more outer layer which presents above your cell wall. It's most abundant in bronze lipoprotein, which maintains the structural and functional integrity of the cell envelope basically. Also in gram-negative bacteria, cross-linkage occurs in direct peptide linkage of amino group of diaminopalmic acid to the carboxyl group of the terminal D-alanine. So this is all about the bacterial cell wall. Coming towards the fungal cell wall. Fungal cell wall is basically made up of three components, that is chitin, glucans, and proteins. So chitins are the polymers which consist of unbranched chain of beta-1,4 linked N-acetylglucosamine. Chitins are synthesized and extruded at the plasma membrane. Coming towards glucans. So glucans are nothing but a glucose polymers that functions to cross-link chitin polymers. Beta glucans are the glucose molecules linked via beta-1,3 or 1,6 bond and provides rigidity to the cell wall, while alpha glucans are defined by alpha-1,3 or alpha-1,4 bond that functions as a part of matrix in this glucan chains. Proteins, these are nothing but the necessary enzymes for the cell wall synthesis and lysis Also, they are also a part of structural proteins which are present on the cell wall. Most of the structural proteins found on the cell wall are glycosylated and contain mannose. Thus, these proteins are also called as mannans. So, I have taken an example of one of the fungal cell here. So, this is a mannans, or these are the proteins. Then this is the glucan chains, and this is a chitin membrane. So basically, glucan and chitins are composed of primary cell wall. Proteins are composed of secondary cell wall. The outermost surface of the cell wall, which provides a medium between the cell and the environment. Also, it acts as a site where the antigen and agglutinin gets attached to the substrate host and other cells. So it will also helps to attach to the host cell basically. So this is all about the fungal cell wall, and the next is our plant cell wall. Plant cell wall, it differentiates a plant cell basically from the other eukaryotic cells. The cell wall is tough and provides a definite shape and structure to the plant cells. It composed of these many things like middle lamella. These many layers basically: middle lamella, primary cell wall, secondary cell walls, and tertiary cell wall, as you can see here. This is a middle lamella. So middle lamella, which is found between adjacent two cells and present outside the primary cell walls. So this is present between the two cells. This is middle lamella. Primary cell wall, it found exactly after the middle lamella. It's a flexible and capable of growth and expansion, and also formed by cellulose fibrils. It composed of uh, various things like hemicellulose, pectins, structural proteins, etc. We'll be seeing the structure of primary cell wall in next slide in details. So this is a basic structure of primary cell wall, which contains cellulose, pectins, hemicellulose, xylane, and proteins. So cellulose, which provides shape and strength to the cell wall. Pectins, which is also a group of polysaccharides which are rich in galactouronic acid, rhamnose, arabinose, and galactose. Pectins are also present in high concentration in middle lamella, where they are presumably serves the function of cementing adjacent to the cell. Hemicellulose, these are the matrix polysaccharide, build up a variety of different sugars. They differ in different species and in different cell types. Okay. Xylene, it is a typically makes up roughly of 5% of the primary cell wall, and mostly it is present in secondary cell walls in dicots. The hemicellulose polysaccharide is linked with xylose and arabinose. Uh, Last but not the least is proteins. Different types of proteins are present in cell wall, most of which are linked to the carbohydrates, forming glycoproteins. The cell wall glycoproteins extension contains unusual amino acids, that is hydroxyproline, which is generally absent in protoplast. Secondary cell wall structure, which is extremely rigid and provides strength and found in all type of cells. It composed of cellulose, hemicellulose, and lignin. So it have basically three layers, that is S1, outer layer, S2, and S3. So this is the inner layer, this is middle layer, S2. Next is the tertiary cell wall. Its next layer is the tertiary cell walls. It is deposited in very few cells. It considered to be a dry residue to the protoplast. Besides cellulose and hemicellulose, xylem is also present here. So this is all about the plant cell wall. Coming towards the general functions of cell walls, so they determine the morphology, growth, and development, protect the cells from invasion by pathogens like viruses, bacteria, and fungus. Basically, for plant cells, they are rigid structures and thus helps the plant with withstanding the gravitational force. Plant cell walls helps in withstanding the turgor pressure, which develops within the cells due to high osmotic pressure. And the main function, it isolates the inner environment of the cell from the outer one. I hope the information is helpful to you. So please do share and like the video and do subscribe my channel. Thank you very much.